This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. Now we are learning about life processes, part 8. So already we have learned about life processes, then nutrition, respiration, transportation in animals and plants. Then next, the last topic in this chapter, that is excretion. So based on some special features like breathe, eat, the ability to breathe, grow, eat, reproduce, then to sense the environment and to move. Based on this, things are classified into two categories, living and non-living things. And for maintaining the life, they need to perform some essential processes, which are that nutrition, respiration, transportation, excretion, control and coordination, growth, reproduction, movement. All these are essential life maintaining processes. So in that, we are going to learn about excretion. Okay. So excretion, what do you mean by excretion? Excretion means removal of waste. Removal of waste substances from the body. Okay, so removal of waste from body, that is called as excretion. This waste, you know that wherever it may be, the waste substances that may cause harmful effect. So harmful effects can be created by this waste substances. So many waste they are producing during the metabolic activities. So waste, from where this is coming? From where this waste products are, or waste materials are coming? See, in our body, many reactions are taking place, right? So metabolic processes are taking place. So as a part of that, some waste materials are getting produced. So from where? Metabolic processes or some life maintaining processes. From that, as a part of that, waste materials will be formed. Okay, so these waste material will be formed. So this waste material, when it is accumulates in the body, and that creates harmful conditions to the body. So to avoid this, we need to avoid this, right? To avoid this harmful effects, what we need? Removal of waste substances which are produced in the body. So that need to be removed in particular time. So on time, this should be removed from that particular organ or where it is getting produced. So based on their accumulation, the body may face some problems, like it may create some harmful effects, like even the organism may die. Okay, so to avoid such conditions, that waste material removal is must. Okay, so for that, excretory systems are helping. Excretory systems are present in our body. They are helping for the removal of waste from the body, which is creating harmful effects, or their accumulation may cause some harmful effect. So to avoid that, this waste material before accumulation, that should get removed on time. Okay, so next we are going to learn about human excretory system. Okay, excretion in human being, how it is taking place in human beings, how excretory system is working. Okay, so human excretory system that is composed of a pair of kidneys. Okay, so human excretory system is composed of a pair of kidneys. Okay, here you can see kidneys. Then from kidney, what is there? Next, a tube. This tube. See, from this kidney, some tubes are arising on both sides. That is ureter. Okay, so that tube is called as ureter, which comes out of this kidney. And where it is going? It is going to the urinary bladder. Urinary bladder. Okay. Then from urinary bladder, it is, or the urine is expelled out through this urethra. Here you can see urethra. Okay. So this is together performs the function of human excretory system. So this is doing the function of human excretory system. What and all there? Just you need to draw this picture, this one, and label it neatly. Pair of kidneys, then the tube which is coming out of that each kidney, that is ureter, then urinary bladder. In that, this tube ends, and from urinary bladder, the opening which you can see, that is urethra. Okay. Next, about kidney. Let's see this kidney. So kidney, you know that it's a bean-shaped structure. It is having bean shape, right? And bean-shaped organ, and this, where it lies, it's near to the vertebral column in the abdominal cavity. Near to vertebral column in the abdominal cavity. Okay. And here this kidney, it is composed of many filtering units. Okay. Composed of filtering units. Okay. These filtering units are called as nephrons. Nephrons. These filtering units are called as nephrons, which are the functional unit of kidney. These nephrons are the functional unit of kidney. Okay. So what we can say, kidney, it is a bean-shaped structure and it is near to the vertebral column in the abdominal cavity. And this kidney, that is composed of many filtering units which are called as nephrons. These nephrons are the functional units of kidney. Clear? So let's see the structure of nephron. Okay. So here in this kidney, many nephrons are there, which are working as a filtering unit. Okay, so here this is the detailed structure. See here, in one kidney, that portion is enlarged. This portion is enlarged. That means a nephron. You can see very clearly here. Actually, Here you can see nephron. Actually, this is, here you can see mess of tubes. Here you can see, right? And there is a filtering part here. There is a filtering part which is called as glomerulus. Filtering part, glomerulus. Okay, so this glomerulus, here you can see it's clearly marked glomerulus. This is a network of blood capillaries. Glomerulus. Network of blood capillaries. Okay, so to this renal artery that is attached to this glomerulus, renal artery is attached. Here you can see that. Renal artery which carries oxygenated blood. Okay, renal artery. This picture also you need to draw this figure. Then you need to mark like these portions. Renal artery. So to glomerulus, this renal artery is attached. Okay, so glomerulus, that is a network of blood capillaries. Here you can see that network of blood capillaries, and to that renal artery is attached. And here next point is the artery which is taking blood to that glomerulus. Taking blood to glomerulus. Which is that artery? That is called as afferent arteriole. Afferent, see, afferent arteriole. This is the artery which is taking blood to glomerulus. So afferent arteriole that is taking blood to this glomerulus. Okay, and the one which is receiving blood from the glomerulus, that is called as afferent arteriole. Okay, here this is receiving blood from where? Glomerulus. Receives blood from glomerulus. That is which one? Afferent arteriole. So which is giving blood to glomerulus, that is afferent arteriole, and afferent arteriole is the one which is receiving blood from glomerulus. Okay, and here you can see this glomerulus, this portion that is enclosed in a capsule like portion. Glomerulus is enclosed in a capsule like portion which is called as Bowman's capsule. Bowman's capsule. Okay, so this is Bowman's capsule. So this capsule like portion where this glomerulus is enclosed, this is glomerulus, it is enclosed in a capsule like portion which is called as Bowman's capsule. Bowman's capsule. And here next you can see this Bowman's capsule extends into a fine tube. Here you can see that Bowman's capsule extends into fine tube, and that tube is highly coiled. This is a highly coiled and Okay, here you can see this is extending into a highly coiled tube. Then next tube, that tube from different nephrons, that is converging into this collecting duct. Okay, and that finally where it goes to ureter. Next, let's see filtration in glomerulus. Filtration in glomerulus. Okay, see how this filtration is happening. It is due to very high pressure. Due to very high pressure inside the glomerulus. Inside the glomerulus. Because of that, this filtration is taking place. Okay, and the lumen of this efferent arteriole. See, this is the efferent arteriole. That lumen, efferent arteriole lumen, that lumen is smaller than the lumen of afferent arteriole. Is smaller compared to the lumen of afferent arteriole. This afferent arteriole lumen is smaller. Okay. So because of that, see the outlet or the lumen through which it is going out, that lumen is smaller. Okay. So due to that, the blood which is entering to this glomerulus, entering, that experiences very high pressure. Okay. So the efferent arteriole lumen is smaller than the lumen of the afferent arteriole. Okay, that because of that, here the blood which is entering into the glomerulus, that is experiencing very high pressure. Okay, so due to that high pressure, this waste products are filtered out through the membrane of capillaries. Waste product filtering. Waste products are filtered through the capillary membranes. Okay, so the capillaries which are present in this glomerulus, through that membrane, filtering of the waste product that will take place. Then this filtered blood, here this filtering over, right? So this filtered blood, where it will go? This filtered blood is sent to the systemic circulation. Systemic circulation. And again through this efferent arteriole, it is going. It is circulating through this efferent arteriole and filters through the, and that filtrate. Here efferent arteriole, again it is going there. Here filtration is happening, then it is moving. After filtration, it is going to the efferent arteriole and to the Bowman's capsule. That's the procedure of urine formation. Okay, that's how urine is formed, where inside the kidneys. Okay, then in the tubular part, here you can see the tubular part of this nephron. There also some reabsorption procedures are taking place, or some processes are there which are helping for the reabsorption. Okay, reabsorption of water and some other filters. Okay, so other filtrate which is taking place in the tubular part of nephron. Here you can see the tubular part of nephron, their reabsorption of water and other filtrates that is taking place where in the tubular part of nephron. So that due to that reabsorption, that increases the concentration of urine. That reabsorption increases, again water is getting absorbed, right? So the substances or the that filtrate concentration is increasing. Urine concentration is again increasing because little more water got reabsorbed from that. So concentration of urine is increasing. And human urine that is composed of water and urea. Okay, so that's how filtration in glomerulus is taking place. The first filtration happens because of very high pressure inside the glomerulus. Then I said lumen of efferent arteriole, that is smaller than that lumen of efferent arteriole. So due to that difference in lumen size, blood which is entering to the glomerulus experiences very high pressure. Okay? And due to that pressure, the waste products are filtered out through the membrane, capillary membrane. So capillary membrane, that is a thin membrane, right? So through that membranes of the capillaries which is present in the glomerulus, that filtration is taking place. Okay, so the filtered blood, now filtration is over. So after that filtration, that is sent to the same circulation through the efferent arteriole, and that filtrate, where it will go? Filtrate will go to the Bowman's capsule. Okay, so that's the way of urine formation. That's a procedure of urine formation. And in kidney, this is how urine is getting formed. Then again in the tubular portion, there again some water absorption or reabsorption of water and some filtrate, some other substances, that is also taking place where in the tubular portion of nephron. So again reabsorption is taking place, right? Water content is reducing, so that increases the concentration of urine. Then here human urine, the last point, human urine, that is composed of water and urea. Okay, this is the excretion in animals, especially we are explaining about this human beings. Next, what about excretion in plants? Excretion in plants. See here, you need to learn the structure and function of nephron, how it is functioning. This nephron, that unit, that means urine formation, that procedure you need to, or that whole, I think first you need to draw the structure of nephron, then you need to mark everything neatly, then write about each portion and the function of each part. Okay. Then another question from this part is excretion in plants. Okay, so let's discuss first excretion in plants, how it is taking place. Actually, in plants, there is no special organs, no special organ for excretion. Okay, for the removal of waste, there is no special organ, and this waste product of respiration, there are also some processes like respiratory process is going on, then photosynthesis is going on. For all these, some chemical reactions will take place. So after this also waste products will form. So waste product of respiration and photosynthesis. Of respiration and photosynthesis. That are used as raw material for each other. See, respiration and photosynthesis, that waste of respiration and photosynthesis, they are used as raw materials for each other, right? Then oxygen, that is produced as a byproduct of photosynthesis. Oxygen, byproduct of photosynthesis. Here also waste materials are getting produced, but that one or the other way, that is also getting utilized for the other procedure or the next process. So oxygen gas, which is produced as a byproduct of photosynthesis, that is used during respiration, used for respiration, right? Then carbon dioxide, which is produced by plant respiration, that is used for photosynthesis, right? So here, whichever waste material or byproduct which is formed during respiration and photosynthesis, that is utilized for the next process. Like oxygen, which is produced during photosynthesis, that is used for respiration. Okay, so that part is clear. Then carbon dioxide, during respiration, carbon dioxide will get produced. So that carbon dioxide is utilized for photosynthesis as the raw material, right? Then in different ways, this excretion is carried out in plants. See, let's see in which and all way the excretion process is taking place in plants. Actually, gaseous waste. The waste materials which are produced in plant bodies in the form of gas, which and all they? Oxygen, carbon dioxide, then water vapor. Water vapor. All these are removed through stomata of leaf. Stomata of leaves. Okay. Then lenticels of stem. So through these parts, stomata and lenticels, through that gaseous wastes are expelled. Then some other type of waste, some waste products that are collected on bark of trees and on leaves. How these will get removed? When leaves and bark, they are shedding. During that shedding process, these waste material leaf and bark, that time these wastes also may get removed. And some other types of waste, they may be stored in the plant body. Why that waste material is stored in the plant body itself? Because that may not cause any harmful effect to that, or that is harmless to that plant body. Okay, like tannin, resins, like secondary metabolites, gum, resins, tannin, then gum, all these are not causing any harmful effect to that plant. So that may get stored on the plant body itself. Okay, so these are the ways of excretion in plants. Okay, so here we'll get another question like, what are the methods used by plants to get rid of excretory products? Okay, so these 3 points you need to mention with example. Okay, and so here in part 7 and 8, double circulation you need to learn. What is the importance of double circulation? Then as a flow chart you need to make the flow of blood in heart. Then importance of double circulation you need to learn. Then xylem and phloem transport, in that what is the difference, that you need to learn. Then structure, here in the 8th part you need to learn about the structure of nephron and function. So these are the main questions in the NCERT book. And next, in your NCERT book you can see many activities. Okay, so just discuss these activities. So let's start with the first one. See, first activity in this chapter, while learning about photosynthesis, there, to confirm that light is essential for photosynthesis, we need to do some experiments, like for what and all materials are required. First, a potted plant with variegated leaves. What do you mean by that? Like some plants are there, like leaves with green and non-green areas. Like suppose here one leaf was there, here some part may be green. Symmetry will be there, and some part, almost every part will be green, and like on the sides and all you may get some white spot or something like that. So like that, those type of plants you need to select. Okay, like croton or money plant and all that leaves. Then we need a petri dish, then tripod stand, wire goes, blotting paper, Doppler spirit lamp, then water bath, iodine solution, butter paper, soft pencil, everything we need to make ready and keep. Then you start. First, keep the things which are all required for the experiment. Then you start the experiment. First, keep that plant in dark room for 3 days. Okay, first, that procedure we are going to start now. So which are all steps we need to follow to do this? First, we need to keep the plant in a dark room for 3 days. Okay, why we are keeping that plant in dark room for 3 days? That starch that should get used up. Whatever starch stored in that leaf, that should get used up. If you are keeping in light, again photosynthesis will continue. So to avoid that, first we are keeping that in a dark room. Then that destarched plant we need to keep in sunlight about Keep the plant in sunlight. Next, first the plant in a dark room. Keep the plant in a dark room for 3 days so that all the starch that gets used up. Then after that, keeping the plant in sunlight about 6 hours. So 6 hours you are keeping. So photosynthesis will start. Right. Then next step is pluck a leaf from the plant. So the same activity which they have given in NCERT book, the same sentence. I didn't change the sentences also. Same thing I have copied here because to avoid the confusion, the same thing I'm going to explain you. Pluck a leaf from the plant. Okay. Then if you're plucking that leaf from that plant, that leaf must be a variegated leaf. Okay. Then mark the green areas. You need to mark the green areas and trace them on a sheet of paper. So what you will do, pluck the green leaf from that plant. That should be a variegated leaf. Then place that butter paper over that leaf. That translucent butter paper you need to keep on that leaf. Then mark the green and non-green areas on that. See, we can make a separate diagram of leaf which is showing the pattern of variegation. So colorless areas that indicate the absence of chlorophyll. Okay. In that colorless areas means no chlorophyll in that area. Okay, so colorless areas indicates the absence of chlorophyll. Okay, then after that, dipping this leaf in boiling water for few minutes. Dip the leaf in boiling water for few minutes. That is next step. Then next step, after this, take the leaf out and now dip it in the beaker which is containing alcohol. Next, you are going to dip that in a beaker which is having alcohol, which is containing alcohol. Okay. Then carefully we need to place that beaker in a water bath and we need to boil. Heat that till the alcohol begins to boil. Okay. So when it is getting boiled, that up to that level we need to heat. Okay. So here we need to take care because highly inflammable alcohol is a substance which is highly inflammable. So we need to take care. Then what will happen? What happens to that color of the leaf in this step? Here, alcohol, for this step we can give the answer alcohol decolorizes that leaf completely. It will decolorize leaf. Okay. Then take out that decolorized leaf. Then we need to dip that in the water for what to remove the alcohol. Then spread that leaf in a petri dish and you can put few drops of iodine solution. Okay, diluted solution of iodine you can put on that, and after 2-3 minutes that excess iodine solution we need to rinse off. Okay, now here you can see before starch test, that leaf was like this before starch test. After starch test you can see this, the second figure. Okay, so the leaf after treatment with iodine, that shows two types of patches. Here you can see two types. Which are they? Some areas are in blue color. Here you can see blue color. but some areas bluish black. Then yellowish part also you can see. So this bluish black areas, that shows the presence of starch. This shows presence of starch. Okay. And this yellowish area, that shows absence of starch. Okay. So from this, it's clear that that bluish green areas, that are the ones which were previously green, right? This green areas that turn to bluish color. Okay, so the non-green areas, that here it remain colorless, or you can see in yellow color here. Clear? So how we can conclude this experiment? Only the chlorophyll containing areas produce starch, right? With the help of chlorophyll, photosynthesis will take place, and that provide the chlorophyll. From this, we can confirm that chlorophyll is necessary for photosynthesis, right? And their photosynthesis will take place, and that shows this blue, that gives the blue color after iodine test. Clear? So this is the first activity, that very simple activity. You can do it very easily. Next activity, second one. This is, so first activity, that shows light is for photosynthesis. Light is essential for photosynthesis. Right? Through this experiment, we can prove that. Second activity, this is to prove that carbon dioxide is essential for photosynthesis. Carbon dioxide for photosynthesis. How we can prove this? See, for that, first you need two same-sized healthy potted plants, then two bell jars, two glass slabs, then Vaseline or grease, something. Then potassium hydroxide, watch glass, then iodine solution you need to make it ready, then water bath, alcohol, then petri dish, spirit lamp you need, then tripod stand, matchbox, all these things you need to make it ready. Then you need to start. Okay, so first you need to take two healthy potted plants which are nearly the same size. Okay, keep them in a dark room for 3 days. Then place each plant on a separate glass slab or glass plate. See, on a separate glass slab or glass plate, you need to place each plant separately. Then, after that, potassium hydroxide. Potassium hydroxide in a watch glass you need to take and place this watch glass where in one of the plant. Here you can see in the first figure, potassium hydroxide is placed in that watch glass, right? Why we are placing this potassium hydroxide? Potassium hydroxide that can absorb carbon dioxide. Okay, so here potassium hydroxide which you have kept in the first plant, that can absorb carbon dioxide. Then both plants you can cover with separate bell jars. Here in the figure it's clear, separate bell jars. With that you are going to cover the plants. Okay, then to seal that you're using Vaseline or grease, any one of them. With that you can use airtight condition. Okay, you can make airtight condition. Then keep these both plants in sunlight for 2 to 6 hours. 2 to 6 hours you need to keep that in sunlight. Okay. After that, pluck a leaf from each plant. Okay. Then same test which we have used for the presence of starch. In both you need to do this separately. So what we can observe here, the presence of starch that can be tested. Through which test? Iodine test. Again iodine test. The leaf of a plant which was kept without potassium hydroxide in that normal photosynthesis. So in this normal photosynthesis will take place because there is no potassium hydroxide. Okay, so that got all the materials which are all required for photosynthesis, light, carbon dioxide, everything that got. But with potassium hydroxide, because potassium hydroxide absorbs carbon dioxide, so the plant with potassium hydroxide, there no photosynthesis occurred because first of all it was airtight setup, right? Experimental setup was airtight. So carbon dioxide which is present inside the bell jar, that was absorbed by the potassium hydroxide. So the plant could not make starch in the absence of carbon dioxide. Here, carbon dioxide got absorbed by potassium hydroxide. That in the absence of carbon dioxide, photosynthesis will never take place. So here, carbon dioxide is necessary for photosynthesis. To prove that we are doing this experiment, so this experiment shows that carbon dioxide is essential for the process of photosynthesis. Okay, next activity, that is the iodine test for the starch presence in the leaves of green plants. This is a simple test. Presence of starch that we need to confirm. So this iodine test is performed to detect the presence of starch in any substance or any compound. Iodine test will be done to confirm that. So this iodine solution which is added to that starch, what color it will give? It gives a blue black color. Iodine solution plus starch, what it will give? It will give a blue black color. Okay, so this is called as iodine starch test. With the help of this test, we can confirm the presence of starch. So let's go to the procedure. First, this money plant or something we can use, take that green leaves and boil that leaf for 10 minutes in water. Okay, and then boil the leaf in alcohol. So how you will boil that? So that beaker which is containing alcohol, that should be placed in the water bath. Don't go for direct boiling. So in water bath, you should keep that. After that process, what will happen? That leaves will become completely free of chlorophyll. Then you need to wash the leaves. Now place the leaves in iodine solution. Okay. So after few minutes, what you can see? That leaves will turn bluish black color. Means their presence of iodine is confirmed. Next test is, that is a starch test, simple test. The next is for carbon dioxide. Okay, here carbon dioxide, that is exhaled during respiration. That we are going to prove. So here we need two test tubes, A and B. You need to mark it as A and B. Then one with lime water. So first, what we are doing is freshly prepared lime water. See here you can see the lime water. So first, freshly prepared lime water in a test tube you need to take. Then blow air from the mouth into this lime water. How you will blow? With the help of a glass tube you can blow. Then in another test tube you need to take same amount of lime water in another test tube. Okay. Then here, with the help of air pump, you can blow air into this lime water. Okay. So what you can see here? See, using a syringe or something, or air pump water, maybe you can use that and blow air to that. So in that test tube, second one, in this one, lime water turns milky. Turns milky. But here, no change. It is as such. So what you can learn from this, lime water turns milky only when carbon dioxide is mixed with it. So here, carbon dioxide is there, right? So that experiment, that shows that exhaled air that contains carbon dioxide, and that proves that carbon dioxide is producing during respiration. So that there is a little amount of carbon dioxide in the fresh air here. From fresh air, very little amount of carbon dioxide is entering there. So that is not turning that lime water milky. Okay, here when exhaled, that air contains more carbon dioxide, which is helping that lime water to turn milky. Okay, so like that we can conclude. That's the conclusion. You need to write both points. Okay, what is happening to the first tube and what is happening to the second tube. You need to mention both. And next activity is to find the product which is produced by the fermentation process. Fermentation. Okay, so here we need to take fresh juice or some sugar solution. in a test tube. Then we need to add yeast to that. Next step is we can put a drop of non-drying oil over that juice surface so that there is no contact remaining in the air. Okay, there is no contact with the air and that juice which is remaining in that tube. Then with a single hold cork, one hold cork, we need to cover the mouth of this test tube. There should be a hole. Okay. And one tube, that delivery tube over the mouth of this test tube we need to keep, and the end of the tube should not touch the fruit juice. Okay. Then other free end of that tube, that should be dipped into another test tube which is having a freshly prepared lime water. Clear? So what you can observe here, after some time, that mixture, that means juice plus yeast, that mixture, that becomes frothy. Okay, so bubbles of gas that is coming out of that, you can see the bubbles of gas which is coming out, and that is passing to that lime water. So that's why it is turning milky after some time. Okay, then you need to remove that cork and just smell the solution. You can experience the smell of alcohol. That alcohol smell will come out of that. See, yeast. So what we can, conclusion, you need to write each and every activity. After that, you need to write the conclusion. This yeast, which kind of respiration is going on there? Anaerobic respiration. So in the absence of oxygen, there is no oxygen. So in the absence of oxygen, anaerobic respiration will go on. And this process of fermentation, there alcohol will get produced and the carbon dioxide is getting released. Here, anaerobic respiration that you need to mention. Then fermentation. Then alcohol production. See, lime water is turning to milky. That is because of the release of carbon dioxide. Okay, what does this tell us about the product of fermentation? And when we are removing the cork and shaking, you can make out the presence of alcohol. That smells like alcohol. Okay. Next is a very, very, very simple experiment or simple activity. Aquarium. Most of the people will be having a simple aquarium in their home. So this is to observe the rate of respiration of aquatic animals. Rate of respiration in aquatic animals. This even small kids can do. So we need a simple aquarium. You know that terrestrial animals, they can breathe with the oxygen which is present in the atmosphere. But what about the aquatic animals? They need to use the dissolved oxygen. Okay, so the amount of oxygen dissolved in the water, that is fairly low when we are comparing the amount of oxygen which is present in the air, right? But we know that in aquatic animals and terrestrial animals, respiration will be there, but respiratory organs are different. And in fish, here we can see that fish respiratory surface, or how respiration is taking place with the help of gills. So due to the unique structure and the features, gill slits and others, right? So due to that features, the gills, they have a large surface area, right? Large surface area they have. Okay, and the gills, they are very thin, right? And they have very good blood supply. Okay, so why these fish, they are opening their mouth and gills frequently to meet their oxygen demand? So your first question is, are the timings of the opening and closing of the mouth and gill slits coordinate in some manner? So we need to write what answer we'll give here. First, yes, during breathing, fishes open and close their mouth, right? Have you observed? They will close and open their mouth and gill slits. So that is also in a coordinated way, and timings also they are maintaining, right? Yes. Next question, count the number of times the fish open and closes its mouth in a minute. See, based on the type of a fish, if it is a large one or a small one or a specific type, it depends upon the type. Upon type of fish, right? Next is, compare this to the number of times you breathe in and out in a minute. See, dissolved oxygen in water, we know that it is lower than the oxygen which is present in the air. So the rate of breathing, that means breaths per minute, that will be faster in aquatic organisms compared to the terrestrial organism, right? But the normal respiratory rate in a adult human, that is around 12 to 16 times per minute in adult human. Okay, but in aquatic organism it will be more, because dissolved oxygen in water, that is less compared to the free oxygen which is present in the atmosphere. Okay, so the rate of respiration in aquatic animals is higher than the terrestrial animals. Okay, so this is a very simple activity. Next is to find out the range of hemoglobin in human beings and animals. Okay, so according to this activity, first you need to visit a nearby health center to find out the range of hemoglobin. So this hemoglobin, where it is present, that is found in the RBC, and that carries oxygen from lungs to other parts of body, right? Oxygen carrying pigment in blood, that is hemoglobin. And lack of hemoglobin that causes anemia. right? And the person who is suffering from that anemia, they looks like pale and they are very weak and pale, anemic patients. So we need to take iron rich food to increase the level of hemoglobin. Okay, so here the questions are like, in children up to 12 years, hemoglobin 11 to 16 grams per deciliter. Then in adult, hemoglobin range is 12 to 18. Then female, in female 12 to 15, male 12 to 18. This and all, nowadays blood test is common, so you can see in that the range and all. Next, here the question is calves. In calves, what will be the range? 10.5. Okay. In cow, 10 to 15. In buffalo, 12 to 14 grams per deciliter. Then we know that human body that need more oxygen to do all the biological works. So here we can say the range of hemoglobin in human being and other animals, that varies. How it varies or what is the reason? Due to different reasons, like that may be hormonal reasons or hormonal changes, or from species to species it changes. Then based on age, all these are the reasons. Okay, next, the last activity, that is to find transpiration in plants. This is based on transpiration in plants. Okay, for that, first we need to take 2 pots filled with soil, one with plant and another without plant, one with plant and one without plant. Okay, then in the second pot just put a stick. Okay, in the second pot, then cover both pots with a plastic sheet and pour some water in the pots. Okay, then what you can observe, we need to cover both sets. So after some time, we can notice that some water droplets are present on the plastic sheet. So plastic sheet, on that water droplets you can see. Okay, water droplets we can see on the sheet which is covered on the stick. So with this activity, we can understand what is transpiration. We have already discussed what is transpiration. Actually, the loss of water from the aerial parts of plant in the form of water vapor, that is called as transpiration. Okay, so that is mainly taking place through stomata, but through the surface of the leaves, so that also it may take place. Okay, so this last experiment is a simple and easiest one. Just you need to take 2 small pots, one with plant and without plant, and in one you need to keep a stick of the same height, then cover both pots with plastic sheet and pour some water in both pots. Put both pots covered with the plastic sheet for some time. There you can see some water droplets which are present on the plastic sheet. So that shows transpiration. Okay, so here we have completed the chapter life processes. Almost all the questions we have discussed. So wherever MCQs, important MCQs are coming, that and all we have marked, and we have discussed 1 mark and 2 mark questions, and along with activities we have discussed. So hope It's clear now. So in the next class, we'll move to next chapter. Thank you.